Hi, everybody. Welcome. I'm Sharon Anderson, president of Leiomyosarcoma Support and Direct Research, or LMSDR for short. And today we're very fortunate to have Dr. Gregory Cote to discuss his trial with us. Dr. Cote is a medical oncologist and investigator at the Henry and Belinda Timar Center for Targeted Therapies, the Center for Sarcoma and Connective Tissue Oncology, and the Stephen L. Harris Center for Carcinoma at Massachusetts General Hospital Center. He's also the assistant professor of medicine at Harvard Medical School. So just before we begin, a few reminders. Please keep yourself on mute during the presentation. After that, you can unmute yourself and ask questions or put your questions in the chat box. This webinar is recorded and will be available on the LMSDR website. So please don't disclose any confidential information when you're asking questions. Okay. And thank you, Dr. Cote. I'll let you take it away. That rhymes.
Thank you. Hey, everyone. Really nice to meet all of you. Excited to be here and talk about this interesting trial. Let me get my Zoom up here. My PowerPoint. All right. Can you guys see that okay?
Yep, I see it.
Perfect. All right. So I thought what I'd do is kind of talk a little bit about some of the challenges with developing drugs in leiomyosarcoma and really all sarcomas for that matter. Give you a little primer on clinical trials and how these work, because I think it helps put things in context. And then I'll give you a little bit of a roadmap of how I think about patients when we're bouncing from trials and different therapies. And then we'll jump into the trial. Plenty of time for questions at the end. If somebody has a really burning question, it looks like a fairly small group. It's okay I can pause if something's confusing. So jumping right into it. So as you guys know, sarcomas are rare. So they're about 12 to 16,000 cases per year. That's about 1% of all cancers. So, not common. And I think what can be a little mind-blowing when you think about this is they're even within that 1%, there's 50, maybe 75 or 100 different subtypes of sarcomas. And these all have their own unique biology and distinct behavior. And it can be a little dizzying when you're trying to design new therapies and try to target these individual treatments, because these individual sarcomas, because the treatments might be different for one versus another. When we try to break this down into like subgroups, so the current pathology approach is this histomorphologic approach. What the heck is that? So this is where the pathologists look under the microscope and try to see, what tissue does this look the most like? So if you think of that top one, you guys can see my pointer, right? Yeah, the adipocytic adipocytes just means fat. So these are tumors that look like fat cells, could be vascular cells, bone, cartilage, smooth muscles. And so they try to pigeonhole these into different groups. And to take this a step further, each of these actually has its own subgroup. So there's many different types of cancers that fall within this group. Some of these are more benign, some are more aggressive. So leiomyosarcoma falls into this smooth muscle group, and there's a few different tumors in that group. So building on this, when we think about the challenges for designing new therapies for this disease, as I mentioned, they're small numbers. So if you think about more common cancers like breast cancer, lung cancer, hundreds of thousands of cases a year, we're talking about thousands. And even within these subgroups like leiomyosarcoma, some of you may know there are we call a heterogeneous clinical behavior, where some people can have metastatic disease that grows very slowly over time, and others can take off and grow really quickly. In leiomyosarcoma, for most leiomyosarcomas, we're missing some of the targets that we see in other cancers. So some carcinomas like breast cancer, lung cancer, melanoma, for example, they have mutations in these genes that are that lead to these proteins with almost like a gas pedal stuck on. So there's this constant drive to have this tumor grow, and they become addicted to the signal to grow. And the way these proteins have mutated, we can actually target those with specific drugs for those cancers. We're lacking those kinds of drivers in leiomyosarcoma. Most of the time, we see mutations in something called a tumor suppressor, which is almost like the brakes have been taken off the system, and those are very difficult to drug. There are some exceptions, but for the most part, that's the case. And then this is less true now as it was when I started in taking care of sarcoma patients and designing trials. But this has been a lower priority for drug companies. As I said before, hundreds of thousands of cases of other more common cancers. And it's just a big market to develop drugs. So obviously they're going to get the priority. But fortunately, there's been a growing interest in companies for looking at sarcomas as sort of an untouched area to develop drugs. So I think this landscape is changing. And then I think an important point to make is that we really have sort of modest activity, modest benefit for our existing drugs. And what does that mean? It just means it's harder to build off of those prior successes to find new drugs because our current drugs aren't even that great. So taking this together, there's really high interest in trying to find more effective drugs that can make people live longer, can stop these tumors from growing, and then importantly, they're also tolerable, right? We want to make sure everyone has a high quality of life when they're going through this journey. All right, so let's segue a bit into clinical trials. So first off, what's a clinical trial? Some of you may have never heard of this. So this is where we're trying to find a new drug for a particular disease to develop a treatment for cancer. And there's many different flavors for this. I'm just going to give you some kind of high-level examples here. This is the roadmap of a drug when it goes from the lab, so what we call preclinical development, all the way to FDA approval. So when somebody has a particular cancer, particular target they want to go after, a laboratory, could be a university, could be a drug company, will develop a compound that hits that target. They run all sorts of models. So these could be cancers that are grown in petri dishes, could be mouse models, and there's a certain bar that has to be met to say, okay, this is a real drug hitting a real target that's going to be meaningful. If that's hit, then we enter something called phase 1. So these are first in human trials. This is the first time a drug's been given to a person. And the real question we're trying to ask here is this drug safe and what's the best dose? We don't have time to go into all the details how these work, but just conceptually, you can think of it starting at a very small dose, treating a couple of patients, sometimes 3 at a time, and seeing how they do. If it goes well, we get a sense of the side effects. We can increase that dose, treat some more, increase that dose, treat some more, and eventually you learn what the right dose is of that drug. Once you reach that sort of recommended dose that we think it's going to be, we could move into these phase 2 studies. And this is where we're treating more patients with the same dose to try to hit some early signs that it is working. And as this drug matures, we go into these larger trials, which are phase 3, where these are the ones you might read about in the news, where we're comparing a drug versus either a placebo or some standard of care other drug and showing that it's a better. And if it is, then it might get FDA approved if it checks all these boxes. Now, you can imagine this is quite a process. So this could take billions of dollars, could be 10 years or more. So there's enormous pressure to try to find ways to be more efficient in this process. And a lot of smart people are working on that. We use what we call different endpoints. So maybe we don't have to show that a drug improves survival, but maybe some surrogate, like tumors aren't growing, tumors are shrinking, things like that. It also means that for these earlier phase studies, we're really trying to get a signal that it works before we launch into these bigger trials, because we don't want to waste anyone's time. Okay, so getting into a little more depth in clinical trials, if you talk to your doc about a clinical trial, the first thing they're going to show you is this thing called the informed consent. What's this? This is a series. This is a document, series of pages that's written in lay language, so not medical jargon, and it basically tells you what the trial is all about. So this will be, what the drug is, who's funding it, what's required of the doctors, what's required of you, goes through the side effects, the schedule, all that stuff. And you'll have a chance to read it, chance to ask questions. And if it's the right trial for you, you'll sign on and you'll move forward. You may read it and say, you know what, thanks, but no thanks, this isn't right for me. Your doctor's still going to be your doctor. They can't, they're not going to treat you any differently. They're just going to find something else, some other way to try to go after your tumor. So it's an important part that you're not a guinea pig, you're a participant in this trial. Okay, you're a partner with us in trying to find this new treatment. If you sign on, the next step is something called screening. So every trial has a checklist of things that you have to do to be eligible for that trial. is the kidney function okay? Blood counts are okay, the heart function, things like that. So every trial is unique. And they'll kind of go through this list of things that you have to do beforehand. And if you hit all those boxes, then you can participate in the trial if you want to move forward. And then from here, you go on to treatment. Now, there's lots of different ways this could be. This could be a pill, it could be an IV therapy, it could be a combination of both, could be a combination of 2 drugs. And every trial is unique in that aspect. And so that, your doctor and the research team will go through the logistics of that. So the key points I wanted to highlight here, this is informed consent. This is your choice to participate. For sarcoma studies, it's pretty unlikely you're going to get a placebo. Occasionally we'll see this, but for the most part, you're either getting a drug versus some other standard or the drug by itself. For these early phase studies, so that's the first in human and the early phase 2 studies, they tend to be pretty busy, lots of blood draws. We got to make sure you're safe. We have to measure the level of the drug in you. Sometimes there's required biopsies. This is where we're trying to make sure that we're hitting the target. So it's a lot of work for you. And if you participate in one of these early phase studies, you really got to have a careful consideration with your doctor about whether it's the right study for you. The trade-off is you get access to these early cutting edge drugs, which can be pretty cool. For these later phase studies, they tend to be less busy because we have more experience with the drug, and it follows a more routine schedule like you might get for regular chemo. Okay, so why don't we jump into how I think my leiomyosarcoma in particular. So the first thing I like to do is just confirm the pathology if they didn't happen here. Why is that? Well, community. Oh, sorry. Can you guys hear me okay? My internet's been coming in and out at work. I don't know why. So I like to confirm the pathology. So even though a community pathologist may be, phenomenal and look at every amazing different type of cancer and that they see, they probably rarely see sarcomas just by the percentages, right? So I always like to have my pathologists who just look at sarcomas take a look and make sure it's the right diagnosis. Occasionally the diagnosis changes, so it's super important to know what you're treating. I generally recommend consultation with an expert leiomyosarcoma oncologist at least once. Again, community oncologists are amazing doctors. They treat all different kinds of cancers. I don't know how they keep track of the literature, but again, probably not seeing a whole lot of sarcomas. So you want to just make sure that you connect with somebody at some point. I work with a ton of community oncologists who co-manage patients. We text each other, we share scans, whatever we need to do to help take care of somebody. And then I generally recommend genetic testing of the tumor. This is where we're looking at the DNA of the tumor to see what's mutated. For the most point, for most patients, we see things, like I said, these tumor suppressors that are mutated that are hard to target. Occasionally, we find something really important, so it's worth it for that off chance that we're going to find something that we can target. So the next question is there a clinical trial? If yes, we're going to talk about that. If not, then the standard of care is either a doxorubicin or anthracycline-based therapy, occasionally in a combination. We'll talk more about that in a second. Or for the GYN doctors, often they'll start with gemcitabine docetaxel. There's probably no difference in starting with one versus the other. Most patients with metastatic disease will get both of these classes of drugs. So either is quite reasonable. So let's talk about combinations. So doxorubicin's been around for 30 plus years, and that's really the standard benchmark of what we're trying to beat. And there's been a number of studies looking at combinations, like adding things to doxorubicin to try to find a better drug. And so most of these have been chemotherapies. And what's challenging here is that although the combinations can increase what we call the response rate, the number of patients with tumors that shrink, what doesn't change is how long people live, meaning you get one drug and then the other versus both at once, people live the same amount of time. So even though the tumors are shrinking, it's not changing survival. So we got to do a little better there. The other key point is that this combination is a lot more toxic. So you're trading off a real quality of life change there too. So we really need to find combinations that can not only improve that response rate, make people live longer, but also reach that tolerability threshold, because we don't quite have that right now with those combinations. Now, some of you may have already been treated with a combination, or maybe it may be considered. We still use these combinations, but we generally reserve it for patients where we need a big response. So maybe somebody's tumor is like very symptomatic. We need this to shrink right now, otherwise we're going to be in trouble. Or we have one shot to make to under control before things go awry. So we still use these combinations. It's just challenging and not necessarily the best option for every patient for the first treatment. Okay, so moving on. So eventually these drugs will stop working. It's just the nature of what sarcomas are, and we got to move on. And so in that scenario, we come back again. Is there a trial? If yes, we'll talk about that. If not, then we'll go back to more standard chemos. So in addition to the ones I meant, there's this drug Lurbinectedin. We'll talk about that a little more in a second. Dacarbazine, pazopanib, things like that. And then outside of this, for the right scenario, we may add radiation. We may add something called ablation, where the interventional radiologist can freeze or burn metastases, and sometimes we'll even do surgery if we have to. So I'd say for the right scenario, all tools are in our tool belt, and you may come across those at some point. And this kind of moves on, so it's like around and around and around. And the hope is that you can bridge these together, one treatment after another, for either a long period of keeping the disease under control and having a good quality life, or one of these really hits where you have a drug that really works, and that can work for a really long time. And that's the dream for all of us who are treating patients. All right, so let's talk about the trial. All right, so the trial is Lurbinectedin plus Doxorubicin, and I'll go through the gist of this. So Lurbinectedin is what we call an analog, so a new version of trabectedin, also called Yondelis. You may have heard of Yondelis. This is a drug that was originally isolated from a sea squirt. This is part of I believe the story is a NIH-funded screening program where they're looking for marine-derived agents that can target cancers. And this drug is approved for leiomyosarcoma and liposarcoma, so a fatty tumor, a fatty sarcoma. And those that's in the later line of therapy. So lurbinectedin is, as I said, a new version. It's approved for a type of lung cancer called small cell lung cancer. It has a pretty complicated mechanism of action. I won't go into it today, but it seems to bind DNA where it can cause breaks in DNA, try to kill the cancer DNA. And it also probably blocks something called transcription, which is when a gene is turned on and off. And what's nice about lurbinectedin is a couple things. One is it's a little more potent, so we can get a higher blood level with less side effects. And also the route of administration is a little more convenient. So it's given over 60 minutes, whereas trabectedin is given over 24 hours. Now, I won't quiz you on this later, but the monotherapy dose is 3.2 milligrams per meter square. This will become important in a couple slides. Okay, so we've been working on this drug for a few years now. And so a few years ago, we published this trial of lurbinectedin in combination with different chemotherapy arms. And in this study, we had one arm was with doxorubicin, just like the study that I'm about to tell you about, but things were a little different. So we used a dose of 2 milligrams of lurbinectedin, which is an intermediate dose. So I remember I told you the full dose is 3.2 milligrams, and 50 milligrams of doxorubicin, also an intermediate dose. And so with these two intermediate doses, patients could get 6 cycles, and then they went on to a full dose of lurbinectedin. And we saw some activity. So we saw 7 what we call partial responses. What's a partial response? So in clinical trials, standard criteria how we measure measure the size of different tumors, and we kind of add those up, and it comes dimensions, and you get like a total number of millimeters of tumor that we're following over time. And if a tumor grows over a certain percentage, we call it progression. If it shrinks by 30% or more, we call it a partial response. If it disappears, it's a complete response. So that's pretty rare in sarcomas. So we saw 7 out of 20 patients who had this partial response, so it shrank 30% or more. So nice signal of early activity. So we got excited about that. But a couple interesting things happened. So For 8 patients, they completed the 6 cycles and then went on to the full dose of lurbinectedin. So their dose increased. And 2 of those patients had partial responses when we increased it. So let me show you these 2 patients. So this is called a spider plot. And again, these are individual patients, and we're following the size of their tumor over time. And we do this measurement where we follow it over time. And so as you follow along here, you can see the tumors shrank a little bit. And then when they went on the lurbinectedin by itself at the full dose, again, increasing the dose, the tumors shrank dramatically. You can see it drop here. And for this patient, same thing, a little bit of shrinkage and then shrank dramatically. So what does that tell you? So in our minds, we said, hey, wait a minute, maybe we have the wrong dose of lurbinectedin because they didn't start shrinking dramatically until we increased the dose. So that was the setup for this next trial where we hypothesized that could we use a full dose of lurbinectedin with a baby dose of doxorubicin and still treat tumors and maybe have some better tolerability. So that led us into this current trial. So just like I told you about the roadmap for patients, this is a roadmap for a trial. It's called a schema. And I'll take you through this because it can be a little confusing. So the first part was a phase 1 where we're trying to confirm this dose. So we had other doses that we combined Lurbi and doxorubicin to, but this is the first time we gave a full dose of Lurbi with a little bit of doxorubicin. So we had to do this early kind of lead-in study to make sure it was safe. And for that, we picked 2 dose levels. So full dose Lurbi again with a tiny dose of doxorubicin. So 25 milligrams in one cohort. So normal is 75 milligrams, so a third of the dose. And then the next group, the next dose level would be 25 times 2, so 50, so 2/3 of the dose. Again, these studies are very small, so just 10 patients in this particular part of the study. And as I told you before, we treat 3 at a time. They do okay, we increase the dose, treat some more. And if they do okay, we call that the right dose because there's 2 dose levels here. If there's a problem, we go back down to the first level. So we went through this exercise and again, trying to figure out, is it safe and what's the best dose? And we were able to determine that the first dose level was the safest and best tolerated dose. So full dose of lurbinectedin with a little dose of doxorubicin. So even though we're looking at safety here, we're also interested in, does this work, right? Because we're trying to be confident that we are using the correct dose to go to the next study. So we can look at how patients do on this study. It's really important to do that, in fact. So these are individual patients. This is called a waterfall plot. And again, as I told you before, we can measure how tumors grow and shrink on the study. So if the column goes up, the tumor is growing. If it's going down, it's shrinking. And what you can see here on this side of the screen is that 6 out of our 10 patients cross this 30% threshold of what we call a partial response. So to us, that's pretty exciting for an early phase trial like that. We wouldn't expect to see that. And for this early phase part, there were a mix of different sarcomas, not just leiomyosarcoma participated, but of these patients, we saw 3 that were leiomyosarcomas. So exciting for this disease. I'll let you process that for a second. All right. So not only are we interested in the response, but also the time that patients are on the drug without progressing. We call that a progression-free survival because, obviously we like to see tumors shrink, but I care more about how long this tumor is under control and not growing. And so this is called a swimmer's plot. And these, again, are the same individual patients, but now instead of looking at their tumor shrinking, it's just the time that they're on the drug. And these are days on the bottom. And you can see here that most patients were on over 250 days. Some of these patients were on over a year. 4 of the patients are still on the study right now. So some of them over a year and a half now, one approaching almost 500 days, which is, pretty unheard of for chemotherapy. So, also very excited for this result as well. All right. And then if people like pictures, this is, do my picture not save? There you go, I copied it over. So this is one of my patients, actually saw him this week, who, you don't have to be a radiologist to see this, but he's facing us in this picture. This is his CT scan at start, and you can see a really big tumor. And as he was on study, you can see it shrank. So really remarkable response. Most sarcomas don't shrink like that, but and I suspect that when we expand this trial, this is going to be a rare finding, but still it's nice to see it working. So in conclusion from this early phase study, we had a full dose with a low dose of doxorubicin. It was feasible, it was well tolerated, and we started to see some early signs of activity. So that met our threshold to go to the phase 2 part of the study. And so RP2D just means the recommended phase 2 dose. So we declare that as that first dose level. And now we start the randomized part. Now, this is just for leiomyosarcoma, and patients are randomized. So computer coin flip, I don't have any control over it. And in one arm, they get that combination I just mentioned. Once they reach their full dose of doxorubicin, they go to the lurbinectedin by itself. And for arm 2, they get standard of care doxorubicin. This is what you'd get anyway. And if it stops working, then they go on to lurbinectedin by itself. And what's nice about this study design is that it lets us do two things. So one, we get to ask the question, does the combination versus doxorubicin work better? Number two, it also helps us answer this question, does the combination versus one drug and then the other, like I was telling you about with the doxorubicin ifosfamide story, whether that might be better. Because we may find that it's better to do one after the other than the combination. So that's, a really important part of this study. So this study's open in Boston. It's pending in New York and some of the Mayo sites across the country. They should be opening soon. And we're really excited to get more patients on, and hopefully we'll finish the study later this year. There is a bigger study coming from the company that makes lurbinectedin coming later this year or later next year that's going to ask a similar question using some different dose levels. So if somebody misses the opportunity for this study, that's a possibility for later. Unfortunately, this is geared towards patients in their first or second line setting. So you can't have had prior doxorubicin or Yondelis to be on this study. But I think it's still an important step for patients with new diagnoses. And, we may find that lurbinectedin is going to be worthwhile as a drug by itself as well. So I think at that, why don't I stop? And then I know there's probably a lot of questions because it's a very confusing topic. So fire.
Actually, I was amazed I could follow that.
All right, nice.
Which I really appreciate.
All right, cool.
And I'm glad to see there's a crossover too, that if they fail on the doxorubicin alone, they can get the drug.
Yeah. I felt really strongly about that when I was designing this study because, I almost exclusively do early phase studies where everybody gets the drug. There's no placebos, there's no other arms. And so it's, for me, just to feel good about the study, I really wanted to include that to get everybody access to the drug. Yeah,
I feel good about this study too. Any questions? And if you're shy, you can put them in the chat box. We've got one from Macy. Macy, you're up late. You're in England. Let's see. Amazing. Have you seen any impacts on those that have been on it I lost the chat. Where'd it go?
No, I got it. Have you seen any impacts on those that have been on it for a long period of time? Do you mean like long-term side effects?
Yeah, I'm just interested, particularly those that you've shown for sort of 300 days or so.
Yeah. So people have been able to stay on this drug for quite a long time. It is like a chemotherapeutic. So there's some nausea when it goes in, some fatigue for sure. The blood counts go down, they come back up. For the most part, people have been able to continue on it without needing delays or dose reductions. We've only had a couple of those. Some people are very sensitive to the fatigue and the nausea. We've had a couple of patients have to reduce for that, but for the most part, people have stayed on it for a really long time where it's working. Great.
That's true for Yondelis too, isn't it?
It is. Yeah, exactly.
On a very long time. Yes.
Yeah. So we think it's pretty similar. We don't know for sure will there be any long-term side effects. We don't have enough long-term experience with it yet, but the hope is that we won't.
Okay, we've got another chat question. Why is chemotherapy a typical prerequisite for clinical trials? If an LMS patient didn't have chemotherapy but had some other drugs such as anazapitinib or no drug, but had taken some Avegant therapy post-surgery, can the patient sign up for any clinical trials?
Yeah, so I think it depends on the trial. For this trial, prior chemo is not required. You are allowed to have prior gemcitabine or docetaxel if your doc treated you with that in the adjuvant setting, but we're looking for patients that are fairly treatment naive for this study. There's no restriction for if somebody got pazopanib or immunotherapy or something else.
Great.
For other trials, it's a great question. So sometimes they're trying to have a more homogeneous group of patients. So they're saying, let's try to get everybody in the second or third line. Sometimes if it's an experimental therapy, the FDA or the company, or sometimes these institutional review boards, which are ethical boards, want somebody to have a standard therapy first before they try something experimental that may not work. But it all depends on the study. And sometimes there's ways around that too. Like, if, like I've had studies that require that, but then somebody can't get some of these earlier therapies for whatever reason. And sometimes you can get around it.
So someone could, who's chemo naive, never had a chemo, could enter this trial.
For sure. That's exactly what we're looking for. Absolutely.
Okay, good.
So I have never investigated a trial or anything. I just talked to my oncologist about it. What's the process if you want to get on the list? How do you go about that? With my oncologist, the discussion initially was, let's try the proven stuff before we try something that's not proven.
Yeah. So certainly, reasonable advice. I kind of think of it a little bit differently. Like we can always do the proven stuff later, when we have a chance to do something that's has an early track record of working, and maybe has the chance to have some longer term benefit. That, to me, that seems like the more exciting thing to do first. But it's, you and your doc got to decide like what's right for you, and it's, trials is not always the right thing. If people are interested, so we're have a little bit of a geographic restriction right now, just given.
I'm close to the Minnesota location. I could get there.
Perfect. Yeah. So then you talk to your doc about doing that, or you can we can give Sharon. There must be a way to post the trial link on your website.
I can do that.
Yeah. So there's something called.
I've been doing it.
So this is important to know, even outside of my trial, but there's something called clinicaltrials.gov. It's a database that's searchable. It's not perfect, but if you type in leiomyosarcoma and recruiting studies, it will filter for that. You can look and see what studies are open. It's going to miss things that are early phase studies that will take any kind of cancer or any kind of type of sarcoma, but it's still a good starting place. On that sheet for any trial, not just this one, you'll see at the bottom a contact number, or an email. Sometimes you can reach out to the person there. It's either the doc or sometimes a research coordinator. And then it's a matter of the logistics. So this trial is pretty easy. So I've had people travel from across the country to do it, just who have the ability to travel. Some trials are very intense, like especially these early phase studies where there could be 10, 12 visits in a month, and that can be hard if you live out of state. I generally like people to have somebody at home if they're traveling, that an oncologist that can work with us just for emergencies. But yeah, it's definitely doable.
Okay, we get some more chat questions. Do you see, Vicki? Do you want to just read that yourself?
Hi.
Hi.
Do you see it working for METs to certain areas more than others? Like my sister has uterine LMS with METs to her lungs, and she might have, she just found out she has a fractured L2 vertebrae, and we're not sure if it's caused by the cancer or a fall. But do you see it working good on METs to lungs or anything better than others?
We've had all sorts of different types of tumors and metastases on this trial, and there does not seem to be a pattern of where it might work best. If you'd asked me beforehand, I would have guessed that the smaller the amount of tumor, the better. But as that picture I showed you, that's about as much tumor as you can get in that gentleman who had that tumor, and has responded. So I think for this drug, it seems to go everywhere. We don't, I don't know about brain penetration. Pretty rare for leiomyosarcoma to go to the brain, but otherwise, it seems to be working for all sites that we've seen.
Okay.
So if somebody was going to go on Doxorubicin next, my sister's only had Gemtax. If she were going to try Doxorubicin next, what would she have to lose by going on this trial?
Yeah. So without getting into the specifics of your sister, for anybody in that situation, I kind of feel like not much, because this is what you would get anyway. So you're either going to get the combination or the control drug followed by the lurbinectedin. I think the only downside is the travel if you live far away, right? And so if people can travel and they want to travel for this, and they'd be okay if they got on the control arm and then getting the lurbinectedin later, then I don't see a downside.
So Prasad asks, does the patient need to be in the Boston area? We live on the West Coast, San Francisco Bay Area, yay. No, you don't have
No,
to.
yeah, along that line of thought, yeah, no. So probably ideally the closest spot for you would be if that patient was interested, would be the Arizona site in Phoenix. It's a bit of a shorter flight than coming to see me.
Are you planning to expand it out to the West Coast?
Yeah, we would love to. So we're this is, so I kind of skipped over this part about clinical trials. So there's different sort of ways a trial can be generated and funded. And so for this trial, I came up with the concept and I got funding from this company called Jazz that it has the licensing agreement with the company that makes the drug in Spain. And so they're being very generous. They've given a whole bunch of money to fund this study, but we are very limited in the amount of sites that we could do. So we worked with our friends at the Mayo and Sloan Kettering in New York to try to get different regions in the area. So the farthest west I could get, a site was in Arizona. There is going to be this larger study that's coming down the pipeline. I don't know when that will open. That probably is going to have multiple sites across the country, including the West Coast. But I don't know the exact timing of when that will open.
Would that be your phase 3 type?
Yeah, so it's not going to be my study. So that study is going to be funded by PharmaMar, who makes Lurbinectedin, and they have a lot more resources than I have, so they can open 30, 40 sites if they choose. But yes, I will be involved in that study to some degree, but yeah.
Okay, any more questions?
Got a hand up.
Oh, we've got a hand up. Okay, Macy.
Sorry, I just had a question, which was, has there been any interest on this study in Europe or in the UK by any chance?
Yeah, very much so. Yeah. So I've been working with some friends across the pond, and there is a lot of interest. They probably are going to wait and open that phase 3 study that I talked about. So it's going to be, that design is a little different. I mean, I think they're still finalizing it, but there's probably not going to be crossover. So that's the one catch with that study. But yeah, that is anticipated to open in Europe.
Okay. How long is the current trial going to run?
Yeah, and until we finish it. So it's 50 patients total, 25 in each arm. We're probably in the phase 2 part, we're probably like, I think 14 or 15 patients into it. So we're, I think it's now that we're about to open these other sites, it's probably going to move a little quicker. But I would love to finish this by end of the year, maybe first quarter of next year. But it's a little unpredictable, right? Because you just got to find the right patients and get them through it. But we're hoping it'll be done soon, because we want to get this data out there, right?
Well, we'll help put the word out. Yeah.
Oh, another question here. Would you be able to coordinate with the University of Miami? Yes. So I talked to them early on. They didn't feel like they had the bandwidth at the time to join the study. There is a Mayo Jacksonville site that will be opening. So I realize that's a far distance from Miami, but it's still in state at least. So that will be, I'm sure they'd be happy to see you there.
Okay. Other questions?
If your insurance denied coverage in a different state, how does that work with the trial?
Yeah. Okay. So that's a good question. So how does insurance work with trials? So just in general, the way these trials work in the US is different in Europe. Your insurance will cover what's called standard of care. So anything that's like a routine visit with the doctor, routine blood tests, things like that. Anything that's research is covered by the trial. So if you have a copay or deductible for the doc visits, you still got to cover that kind of thing. Some trials that are sponsored by companies will give you support for travel. Every trial is different, parking, things like that. And because this is a small study, I don't have that kind of funding to cover that. For out of state, it would every situation, every insurance company is going to have a unique situation with their client. I think it would have if they denied it, then, this is why we think it should be done. And then it'll be a bit of a back and forth. Sarcoma is not, unfortunately not like a public health problem. So it's usually like one-off patients with these kinds of scenarios. So most of the time with the right appeals, getting the right people on the phone, we can get around these kinds of things, but every insurance company is different. Some are very hard to work with, some are easy.
Okay, Julie has a question.
How different is lurbinectedin from Yondelis? Yeah. If you didn't have a great response on Yondelis, is that an indication lurbinectedin may not work? Yeah. So currently lurbinectedin is not approved for sarcoma. So it's only approved for small cell lung cancer. So if you tried to get it, it would be what's called off-label, meaning your doctor is trying to give it outside of the FDA approved guidelines, which can be done. Every institution has a different policy on this. And there's like the insurance issue and things like that. Do we think that it would work better? We don't know. So they've never been compared head to head. I have had patients who've been on other lurbinectedin-based clinical trials who it stopped working, and then they went on to Yondelis trabectedin outside of it, and it didn't work. So my gut is if one doesn't work, the other probably wouldn't work, but we don't know for sure.
Anybody else? Don't be shy. You're going to leave and say, oh, I had one more question.
Okay, let's say, for example, somebody could be traveling to the Mayo in Jacksonville. Like, how often would they have to be going there for the trial?
Okay, yeah. So for other trials in general, every trial is different. For this particular trial, it's actually not too bad because there's the initial visit to sign the consent, maybe another visit to do those screening tests I talked about. And then the trial itself is every 3 weeks, the visits, the infusions. So pretty good schedule. There may be extra visits if somebody's not feeling well or if they, for their CT scans. But if everything goes perfectly well, it's every 3 weeks.
I had another question, if that's okay.
Go ahead, Macy.
I was just going to ask, so what does success look like for you now going forward? And are you likely to change things a bit more in terms of the percentages of the drugs?
So for me, I would like to see two things for this trial, and I don't know if we will, right? That's why we do the trial, but I'd love to see that the combination extends what we call that progression-free survival. So the amount of patients that are on the drug, what's the median time till they start progressing versus doxorubicin alone. And I'd love to get that second progression-free survival. So patients who progress on doxorubicin, say after 4 months, when they go on to lurbinectedin, and maybe they can get controlled for another 9 or 12 months. Like that'd be pretty epic if we see that. Are we going to see it? I don't know. That's why we're doing it. I don't know if we're going to see that high response rate that I showed you. That's pretty unusual. And there's been many examples of sarcoma trials where we see that in the early phase studies. We get into the big population and we don't see it. So I'm not counting on that. I'm fine with that. It's that time we're keeping things under control. I think this trial is going to be really important to set up that larger phase 3 trial from PharmaMar. Because in that they're trying to sort out the right dose, as I said, and hopefully this will provide clarity on the right dosing, and then ideally that study will be positive and lead to potentially a new option for patients for their initial treatment.
Yeah, sounds amazing. That's good.
Yeah.
I want to remind people also there are some resources for people who want to join clinical trials. Not a lot, but there's some, and they're on our website under financial resources. But I know Sarcoma Foundation of America has some funds to help people get to clinical trials, and Lazarus does too.
Yeah, Lazarus has been great. Yeah, and I will say, if this trial isn't right, like say this doesn't work out for people who are interested, there are many other people doing some great trials in leiomyosarcoma that are coming down the pipeline. That some of that are already open. So, I think it's worth trying to stay in tune with like what's out there, and every time you're up for like a treatment change, or even before those scans, kind of start thinking about what are these options ahead of time. Like if we show this current treatment's not working, like what's next? And I always kind of like to think about like a couple options just so that we can, be ready if we need something.
Yeah, absolutely. And I tell people, consider a clinical trial first. you know, because you may be able to get a drug that you won't be able to get later on. And you can always use the standard chemos later on if they're going to be there, so to fall back on. So it's an opportunity to try something new. And a lot of these are already mixed combinations with some of those standard chemos. So if you have the standard chemo, you won't be able to get the clinical trial drug.
I have a question. Have
Yeah.
you seen any complete responses where it's like the cancer is kind of gone?
Yeah, so not yet. We've seen some individual, so for those trial measurements I was talking about, so they have these things called target lesions that you're following and non-target. It's just how the radiologists kind of track things over time. So we've seen some individual spots disappear, but not all the tumors. But I hope one day we will.
Okay, another question is,
Yeah.
that young man that had such a shrinkage, might he now be eligible for like some surgery or something too?
Oh, did we lose him?
It was frozen up.
Oh, I don't even see him. Okay, so somebody, while we're waiting for him to come back, Dr. Cote, somebody asked in the chat to confirm if this link is right. I just did. It is the right link. So, oh, here he is.
You gonna drop here or just me?
Where did you
Can you-
go?
I don't know. My Zoom just crashed. That was really weird.
Okay, well, you got in easily.
All right, I missed the question. Sorry.
So someone just asked if this link they just posted in the chat box is correct to your clinical trial, and I checked it, it is.
Okay, cool.
Yeah.
Yeah,
Okay,
so
I had asked that young gentleman that had such a huge shrinkage, might he now be like eligible for surgery to get rid of it or something? Because it shrunk so much?
yeah, his scans are more complicated than that. So not yet. Can you guys hear me okay?
Yes.
Yeah.
Okay, the internet's really choppy here. I don't know what's going on. So he for that guy, no, I did have one other patient who was able to get surgery, which was pretty cool.
Okay, great. That's always the goal.
Yeah, and then Sharon, if you want,
Whoop. Oh no. We're playing hide and seek with Dr. Cote. Hopefully he'll come back here in a second. Prasad, I see you live in the Bay Area, so do I.
Nice, Sharon. I live in Fremont. You
In
live
Fremont, yeah, I'm here in San Francisco.
Nice.
You'll have to join our local group.
Which local group? Sorry.
We have a local San Francisco Bay Area LMS group just to share local resources.
Oh, nice. Is there any Facebook group that I could join?
It's a Facebook group, yes.
Oh, nice. Thanks. I'll search for it.
Yeah. Okay, we may have to say goodbye. We'll give it another second or two.
Thanks for coordinating this, Sharon.
My pleasure. I am a firm believer in clinical trials because every drug that we have for a treatment had to go through clinical trials. And we need more treatments. That's all there is to it. And
Absolutely.
eventually we're going to find one for everybody. You know, that's going to work. Oh, he's at the door. Here he is.
Sharon,
Yeah.
I'm going to head off now because it's quite late where I am, but thank you very much.
Thank you very much for joining us.
hey,
Thanks so
sorry about
much,
that.
Dr. Cote.
That's okay. We were chit-chatting.
My Zoom just died on me. I don't know what's going on. What I was saying is that if you want, feel free to share my email to the group, and I'm happy if there's any candidates to kind of help kind of triage things.
I can do that. And I also put the information in our newsletter that goes out twice a month. So
Great.
people can catch it there too, as well as our Facebook group that has 4,700 people on it.
Hopefully I won't get 4,000 emails.
I don't think so, but hopefully you'll just get the right people at the right time.
Yeah.
All right.
So,
So since you're kind of unstable here, and I think we've asked a lot of questions, I'm gonna go ahead and close the webinar, but we want to thank you so much. This is very valuable information, and it sounds like a very promising trial. So
Right.
we are very, very happy about, promoting this.
Great. Thanks, everyone. It was a lot of fun, and look forward to seeing everyone again on another meeting.
Okay, thank you, Dr. Cote. Bye-bye.
Thank you. Have a good day.
Bye.
Erin Anderson.
Yep.
Hi, we're in the Santa Clara area. So
Oh, are
what is
you?
the Facebook?
Okay, so this Facebook group, are you on the Facebook group for LMSDR?
I think we are. I'm in a couple of Facebook groups, and I don't remember which they are. But
Okay, I'll post it today, the link to the Bay Area one, but it's called San Francisco Bay Area LMS group.
okay,
So you can do a little search on Facebook for us.
I can do that.
And I encourage other people to try to meet local people because it's just amazing. And you'll see we all look marvelous.
Sure.
There's a link up on the chats for that group.
Oh,
Okay.
thank you. There we go. Okay,
Okay.
there you have it.
Okay.
All right, anybody else have any questions?
Mhm.
We're not on that one.
Any comments? Okay. Thank you all for coming. I really appreciate it. And I hope to have some more clinical trial information coming down the pipeline. For some reason, we've got a lot of clinical trials right now, so we're thrilled. LMS is rocking with the researchers. So we're just going to keep it going. And so everybody hang in there, and I wish you all the best. Okay. Bye-bye.
Thank you.
Thank
Have
you
a
all.
good day.
Bye.